Dr. Amit Bhattacharya from Department of Zoology, Ramjas College, University, Delhi, and the title of my presentation today is the Vector Borne Diseases, which is the second part of the presentation. Now, in the first part of the presentation, we discussed about the vector borne diseases, what exactly the vector borne diseases are. Now, we know that the vector borne diseases are a diverse group of illnesses which have infected various places and most of the countries throughout across the globe. Now, there are various vector borne diseases which can be easily find out, like the Zika virus, dengue virus, chikungunya, West Nile, malaria, yellow fever and Chagas disease. So, these constitutes the vector borne diseases, and every year many of these new diseases are also coming up. Now, as I told you, the vector borne diseases constitute a diverse group of illness which constitute the pathogens which transmit these particular infections. They are mainly the arthropod vectors, which includes the mosquitoes, ticks, sandflies, and how they exactly spread? They spread the germs or the pathogens or the conditions from one person to the other person, or from the animal to the humans over here. Now, a person who gets bitten by a vector and gets sick could have a vector borne disease. Now, we come into another interesting vector borne disease which is called as the chikungunya. Now, the chikungunya is basically a mosquito borne viral disease which includes the transmission of the chikungunya virus. So, chikungunya virus comes under the alpha virus genus of the family, and this is a RNA virus. Now, the name chikungunya has been derived from a word in the Kimekonde language which means to become contorted. Now, the chikungunya virus was first identified in the United Republic of Tanzania in the year 1952, and subsequently various other countries reported the outbreak over there, which includes the Africa, Asia, Thailand and India in the 1970s. Now, the disease is occurring in Africa, Asia, and many of the European countries and the American countries are also coming up with these particular infections. Over 2 million cases have been reported since 2005, and chikungunya has now been identified in over 110 countries in Asia, Africa, Europe and the Americas. So, we can see this WHO released map which shows the spread of the infection of chikungunya across the globe. So, we can see the America, Asia, African countries which have large spread of these chikungunya viruses across the various places. Now, we come into the transmission of the chikungunya. Now, the chikungunya virus, just like the dengue virus, is also transmitted with the help of the infected female mosquitoes, and most commonly the mosquitoes involve the Aedes aegypti or the Aedes albopictus. Now, these Aedes mosquito primarily bite during the daytime hours, and they lay the eggs in the container which is present in the standing water. Now, these species feed outside also, and the Aedes aegypti many a times feed indoors also. Now, a mosquito which is not infected with this virus can take a blood meal and get infected with this particular virus, and when it bites another person, the infection is spread over there. So, that is how the transmission takes place. The virus replicates within the mosquito over several days and get into the salivary glands, and once it is biting a person, it gets transmitted into new human host when the mosquito bites them. So, we can see the electron micrographs of these chikungunya viruses over here which are shown. Now, the virus begins its replication in the newly infected person, and it subsequently increase in the concentration occurs, and they start coming up in the blood circulation. Now, this is the structure of the chikungunya virus which is shown over here. Now, the chikungunya virus is majorly transmitted with the help of the mosquito which is the Aedes aegypti. Now, this is not spread from one person to the other person, so it has to spread through a vector only. That is why it comes under the vector borne diseases. Now the virus needs a vector, which means it requires a transportation agent, which is the mosquitoes, which can be Aedes aegypti or the Aedes albopictus. Now the Aedes aegypti is the vector, the main vector that is present and plays a very big role in the transmission of these arboviruses in Americas and various parts of the country. Now, the mosquito completes its life cycle from the egg to the adult in about 7 to 10 days, and the adult mosquito usually lives for 4 to 6 weeks over here. Now, the female Aedes aegypti is the responsible for the transmission of the disease because it needs blood, human blood for the development of her egg and for the metabolism. So, the male is not the responsible, the female Aedes aegypti is the responsible, and the male does not feed on the blood meals actually. So, this is the image of the mosquitoes which are shown over here. So, the Aedes aegypti and the Aedes albopictus mosquitoes are shown over here, which are mainly involved in the spread of the chikungunya virus to the human host systems. Now, how exactly the transmission takes place? So, this transmission is basically a human mosquito transmission which is taking place. So, from the mosquito it gets transferred to the human where the development process takes place. Subsequently, it comes up in the blood circulation, then it is taken up by the mosquito, it gets infected, it then bites another fresh host and the infection goes on. So, that is how the infection or the chikungunya spreads up. Now, this image shows the chikungunya virus pathogenesis, how exactly the mosquito bites and transfers the viruses from its proboscis into the host system over there, and then the virus enters the cell with the help of the surface proteins and the receptors which are present in the host cells, and then the infection or the further development process takes place within the host system. Now, this is a diagram which shows that how exactly the chikungunya virus develops within the human host system. So, it includes the replication, transcription, translation of the viral genome which is present, so that a number of copies of these particular viruses are formed, and many a time these kind of pathogens or viruses, they use the human system for its development. Now, there are various symptoms which are there which have been recognized with the chikungunya, but many a times they are confused with the symptoms of the dengue infection also, because most of the symptoms are overlapping, which includes the fever, pain in the joints also, loss of the appetite, weakness like this one. Now, some of the chikungunya symptoms are shown over here, which includes the muscle pain, headache, nausea, fatigue, rashes, joint pain is often present over there, which usually lasts for few days to a few weeks over here. Now, most of the patients who suffer from the chikungunya infection, they generally recover fully, and in some cases the joint pain remains over a period of time, but subsequently over the years it may get released up. Occasionally, there are cases of eye and nervous system and heart complications which have been reported, as well as there are some cases of gastrointestinal complaints which have been reported in the case of the chikungunya. Now, the chikungunya is detected again by the direct and the indirect methods. So, there are various direct methods which are there, which includes the detection of the virus in the blood circulation by taking the blood samples, collecting the blood samples, then performing the reverse transcriptase polymerase chain reaction or RT-PCR, or the checking up the viral genome over there or the viral antigens. The indirect methods includes the serological test, which includes the IgM, IgG estimation. So, they gives a idea that whether the person was suffering from this particular infection or not. Now, the person's immune system plays a very important role in the virus infection. So, the antibody levels are detected. They can be also playing a very important role in the detection techniques or the diagnostic technique. So, the antibody levels are typically detectable by the first week after the illness onset and can still be detected for about 2 months over there. Now, there is no specific antiviral drug which are available for the chikungunya treatment which is there, or no commercial chikungunya vaccine which is present. So, but several vaccines are underway or under the developmental processes actually. Now, generally the doctors recommend that many of the drugs should not be taken up during the chikungunya, and many of the drugs are recommended, which includes the antipyretic drugs which reduces the fever and optimize the use of the pain medication by administration of the fluids in the case of the patients who report the chikungunya infections. Now, in the chikungunya and the dengue, mainly the doctor recommends that the aspirin or the other non-steroidal anti-inflammatory drugs should not be administered until the dengue or the chikungunya has been ruled out, in order to reduce the risk of the bleeding conditions. Now, if we see the image over here, this image shows the website of the National Vector Borne Disease Control Institutes. So, this is the agency which keeps a check of all these vector borne diseases which are spreading up or which are being reported across the India. So, this is the website. You can just log on to this website. The information about all the vector borne diseases are available. This is maintained by the Ministry of Health and Family Welfare, Government of India, where you can get information about these viruses or the bacteria or the other vector borne diseases as well as the zoonotic diseases. Then you can get the treatment regimes which are available, which are generally recommended, the measures to control the various things also mentioned in this one. Now, we come into the diagnosis of the chikungunya virus infection. So, it includes the detection of the viral RNA detection or the viral antigen detection which is there. The indirect method includes the antibody detection. So, the viral RNA detection is generally done with the help of the RT-PCR, which the RT-PCR we heard many a times during the COVID infection also, which played a very important role in the detection of these viral infections within a patient. Then the viral antigen detection methods which are there, which includes the ELISA method or the immunosorbent or the immunoblot methods. Then the antibody detection method, which includes the serological methods, which includes IgM or the IgG antibody detection, and this gives a indirect indication that the person might be suffering from the chikungunya virus infection. That's why the antibody titer are very high in these particular patients. Now, there are various IgM kits which have been released. One of the kits which I have shown over here has been released by the Defence Research and Development Establishment, DRD of DRDO India. So, the salient features of this particular kit is, this is a indirect antibody ELISA principle. This is based on the indirect antibody ELISA principle. It utilizes the recombinant chikungunya envelope proteins. The time required is generally 2 hours, and it is very easily to monitor up the detection of the chikungunya infection. Now, some of the important strategy for the mosquito control is the prevention of the mosquito breeding, which includes the indoor residual spraying IRS, where there are many indoor residual sprayers which are available, which are easily fumigated or sprayed up in the areas where the mosquito breeding are taking place. Now, apart from that, there are various protection measures from the mosquito bites are also there, which includes the use of the mosquito repellent, wear long pants and long sleeve shirts, use of window screens, use of bed nets, many a time they are referred as the machardani. Now, we come into the next vector borne disease, which is the malaria. Now, malaria is a vector borne disease which is again spread by the infection by the female Anopheles mosquitoes. Now, this is a life-threatening disease many a times, and it is caused by the parasites that infects the human's host systems, the various organs like the liver, erythrocytes are infected, and the transmission takes place. There are 5 parasite species which have been reported in the case of the malaria in humans. Of these, 2 of these species are poses a great threat to the humankind, which includes the Plasmodium falciparum and Plasmodium vivax over here. Now, in the 2022, it has been reported that nearly half of the world's population was at risk of malaria, and there was about estimate of about 249 million cases of malaria in 2022, and the estimated number of malaria deaths stood to 60,000, 6,08,000 deaths in the case of the 2022. Now, most of these cases are coming or reported from the African regions or the South Asian regions over there. Now, the symptoms of the malaria are basically the severe illness or the death. In the case of the infant child under the 5 years, their various symptoms are being reported, and people with HIV or the AIDS infection are at a higher risk because they are immunocompromised. So, we know that whenever a person is immunocompromised, there are high chances that the person may get many of the infectious diseases or the opportunistic disease. One of the opportunistic disease is the malaria, because as the immune system is lowered down, so what happens is these infections can easily occur because they can't be traced out by the surveillance of the immune system within the host system. Now, the various symptoms of the malaria includes the extreme tiredness and the fatigueness, impaired consciousness, multiple convulsions, difficulty in breathing, dark or the blood in the urine, jaundice-like condition, and the abnormal bleeding which occurs. So, these are the various severe cases of malaria. They report these kind of infection cases. Now, every year the WHO reports or comes out with a report which is called as the World Malaria Report. So, this year also they have come up with a report, and it gives a comprehensive overview and update about the assessment and the trends of the malaria control and the elimination across the globe. So, if we see from the report, this is the picture which is showing the spread of the malaria infection across the various parts of the country. So, we can see generally the tropical and the subtropical countries are the one which are infected by the malaria infection. This is basically due to the climatic condition which are optimum for the growth and the development of the malarial parasite, as well as the vectors which transmits these particular infectious diseases. Now, this is a diagrammatic representation which shows that which are the countries which reported over a period of time that they were malaria free or they have eliminated the malaria infection over a period of time. So, there are various countries across the globe which have been instrumental in controlling the malaria infection by using the various strategies which are available, or the WHO recommended strategies, which includes the use of the proper drugs or the medication, as well as the vector control measures, which includes the indoor residual spray or the various other methods which are available. Now, one of the another species have been added up. Initially there were 4 species, but the 5th species have come up, which is many a time referred as the Plasmodium knowlesi. Now, the Plasmodium knowlesi disease infection is also increasing up, and it is adding up to the burden of the transmission of the malaria. Now, Plasmodium knowlesi has emerged as a notable concern in malaria cases, especially from the Southeast Asian regions, which includes the Indonesia, Malaysia and the Thailand region. It has been estimated that about 2,768 Plasmodium knowlesi cases were reported globally, which is a decrease of 24.2% compared to 2021, which was about 3,651 cases. But the Plasmodium knowlesi somehow is a major concern, because the scientists are still to decipher out the exact mechanisms of the transmission and how exactly this is behaving out. Now, the increase in the burden of the transmission of Plasmodium knowlesi poses a unique challenge for the malaria elimination, and there are various regions which are reporting these particular infection of Plasmodium knowlesi. Now, let us come into the life cycle of the Plasmodium parasite, which is the parasite for the infection of the malaria. Now, the parasite completes its life cycle in two major hosts. One is the mosquito, the other one is the human host. In the human host, it is divided into two cycles. One is called as the exoerythrocytic cycle, the next one is called as the erythrocytic cycle. So, what happens is, as soon as the mosquito bites the human host, it transfers the parasites into the blood circulation of the humans. Through the blood circulation, it goes into the liver cell where it divides to form the scission condition, and the exoerythrocytic cycle start taking place. Now, as this exoerythrocytic cycle takes place, the parasites start growing up within the liver cells, but after reaching a proper level, it bursts out because it cannot take any more of these growth of these parasites. So, all these scissions which are released over there, or the ruptured scissions, released scissions, they go on to infect further liver cells in the human body, and many a times they go on to infect the human RBCs or the red blood cells. Now, in the red blood cell, they start developing in the red blood cells, though they are present within a parasitophorous vacuole in the red blood cells where the growth start taking place, which includes the ring stage, trophozoite stage and the scission stage. So, this is called as the erythrocytic cycle. Now, many a times some of these particular developing stages, they develop into the gametes, which are micro and macro gametes, which are then taken up by the mosquitoes, and the next phase of the cycle or the sexual phase of the cycle start taking place in the mosquitoes, which is called as the sporogonic cycle. Now, the macro and the micro gametes, they fuses to form the eukinete oocyte within the things, and then subsequently the division takes place. The ruptured sporozoites are then through the saliva, it moves into the proboscis, and the mosquito, when it goes and bites another human being, the transfer takes place. So, this is how the cycle of the malarial parasite takes place. So, there are various treatment regimes which are present, which mainly affects the transmission process within the human host system. Now, there are various symptoms which have been reported in the cases of the malaria, and many of the symptoms are very severe in the children under the age of 5 years or the pregnant women actually. So, some of the symptoms of the malaria include the extreme tiredness and the fatigueness, impaired consciousness, multiple convulsions, difficulty in breathing, then abnormal bleeding. So, these are some of the severe symptoms which are found in the cases of the malaria. Now, for the treatment of malaria, the WHO has now recommended the artemisinin based combination therapy or ACTs. So, these are the therapies which are the most effective anti-malarial medicines which are available today, and these are being recommended mainly for the Plasmodium falciparum infection, as this falciparum is one of the deadliest malaria infection which has infected globally. Now, the artemisinin combination therapy, it includes two active pharmaceutical agents which acts on different mechanisms of action on the parasites. So, one of the pharmaceutical agent is artemisinin, which is being extracted from a plant which is called as the Artemisia annua. The other partnered drug basically is another drug which infects or affects another mechanism of action on the parasite. So, taking up two drugs in the combination, what happens is they gives the advantage that as both of them are acting on different mechanisms of action, so the parasite cannot evade these particular actions or the malaria treatment. Now, we see the image over here. This image shows the various artemisinin combination therapies which are available in the market, the drugs which are available in the market, and we see the structure of the artemisinin which is shown over here. The structure of the artemisinin, if we see, there is a oxygen ring which is present over there. Now, what happens is this plays a very important role in the mode of action. Now, the artemisinin is extracted from this Artemisia annua plant actually. So, this Artemisia annua plant gives the artemisinin, which is one of the most important form of the artemisinin which is present in the artemisinin combination therapies. Now, there are various regions which are there for the control of these mosquitoes which are present transmitting these malaria condition, which includes the prevention of the mosquitoes from the breeding grounds. The Anopheles mosquitoes, we know it spreads the malaria. So, we can see the image over here, the Anopheles mosquito larval and the pupal stages are developing over here in the stagnated water, and subsequently if they are removed, they can be stopped over there. So, in this part of the lecture, we discussed about two important vector-borne diseases. The first one was the chikungunya, the other one was the malaria, and we discussed about what exactly the virus or the pathogen which is causing the transmission and the infection, what are the modes of transmission, what are the vectors which are involved in the transmission, as well as the diagnostic techniques which are available and the treatment techniques which are available for the treatment of these vector-borne diseases. So, thank you very much for watching. Thank you.